Kia ora and thanks for joining us. My name is Christine Harper and I'm the Stakeholder Relationship Manager at Manaaki Whenua. Sharing the great work from our research programs is a really important part of how we connect with people, and although you're muted for the webinar, we really do want to hear from you. Please put your questions in the chat box as soon as you think of them. We'll do our best to answer as many as possible in the live session, but those that we can't get to, we will collate and include responses when we upload the webinar material to the website. One special feature of today's webinar is that you can't see us. For some reason, the webinar platform is not allowing us to switch our cameras on. So bear with us being voices coming from behind the screen. Paul Scofield is the Senior Curator of Natural History at the Canterbury Museum. His work over the past 20 years has involved understanding the whakapapa and life histories of New Zealand birds, and today he primarily works in palaeontology. He's a research lead in the collaborative 5-year MB Kiwi Rescue research program. Sometimes research really does turn up some unexpected results and connections, and I'll hand over to Paul to tell you all about it.
Kia ora koutou, everybody. Thank you for this opportunity to talk about this research, which I've entitled this piece of research, Tokoeka Kiwi, a story about a name. Now, names, of course, have a great deal of significance to everyone. Your personal name. To Maori, names have a great deal of significance. But in science, a scientific name is a crucial part of the equation when you're trying to understand speciation. What is a species? What is a unit that needs to be conserved? And it's also important when you're trying to understand the evolutionary pathways that an animal or an animal group have actually gone through. When an animal is first described by science, it's given a scientific name, and the scientific description today contains a great deal of information about why you consider the animal to be distinct, and it's also required to give a type locality and also a type specimen. This method of scientific nomenclature has evolved over the last 200 years, and during the period that I'm going to be talking about today, there wasn't a need for a type locality to be given, and there wasn't even a need for a type specimen. So if a single type specimen of an animal is being described, that specimen is known as a holotype. And the story today I'm going to tell you is about a holotype. And a holotype, it's important to understand, owns the name of a species. So the funded work that I'm going to talk about today is a very small part of the Kiwi Rescue programs. The Kiwi Rescue program was funded by central government, and the genetics program that I've been involved in has the rationale that the Department of Conservation and iwi require a formal publication describing essentially telling everyone two things: how many different types of kiwi there are, and how distinct they are, and how much money, I guess, is needed to ensure the genetic diversity of kiwi in Aotearoa. The two questions I've been trying to address are what is the status of the Tokoeka populations? Are they species or subspecies? And also we've been looking at the same time at the North Island brown kiwi populations. How distinctive are they, and what level of protection is needed to ensure genetic diversity is conserved. The team involved in this piece of research have involved several people from Manaka Whenua, including John Innes, Jamie Wood, and Lea de Nascimento. At the University of Canterbury, my wife, Vanessa de Petrie, has been an important part of the team. At Te Papa Atawhai, we've had Hugh Robertson and Rogan Colburn, and working at the University of Toronto has been Jason Weir. This project has been supported and guided by Tangata Whenua, with iwi and hapu, the hold manawa whenua over Kiwi breeding areas having an active involvement. An annual hui have been held with iwi partners to get guidance and recommendations for the future direction of this kaupapa kiwi. The work that we've been doing has been especially guided by a pivotal piece of research that came out in 2016 that involved a great deal of sampling from all over Aotearoa and established a framework for our understanding of Kiwi diversity. This work was led by Jason Weir of the University of Toronto, and he established that the traditional taxonomy of especially brown Kiwi was fundamentally flawed, and that this idea that there was a single species in the North Island and a single species in the South Island was quite wrong. And in fact, there was a quite high degree of diversity. For example, there were at least 4 living populations of Tokoeka that were quite distinct, and at least 2 extinct populations. And that roe, the kiwi that occurs in only in Kōrero today, probably occurred right through the northern South Island and even into the Wellington area. And the what we used to call the North Island brown kiwi, Mantelli, has at least 4 quite genetically distinct populations. If we now take just the Tokoeka, we find that there are 4 quite distinct populations. The Haast Tokoeka, Northern Fiordland Tokoeka, and the Northern Fiordland population is sister to the Stewart Island and Southern Fiordland populations. So on this diagram, you can see where each live animal was sampled by blood to establish which population it belongs to. And the red arrow there is important. That's Dusky Sound, and we'll get to that in a second. So Kiwi obviously have been known by tangata whenua for more than 1,000 years, but the story that I'm going to tell you today involves the European discovery, quotes unquotes, of Kiwi. And incredibly, it involves this man who was a pirate. Captain Andrew Barclay was a ship's captain in the late 18th and early 19th century. During the Napoleonic and warring periods in early European history, he actually received letters of marque from George III. Letters of marque were basically legalized piracy. And for more than 10 years, he operated as a pirate on the South Seas. He wrote an account of this, which has actually been used by people such as Hollywood, to get an idea of what piracy was like in the early 19th century. And an interesting fact was that when he retired, he actually retired to northern Tasmania and became the first European settler in Evandale, which is near Launceston in Tasmania. And this is the house that he built in the 1820s in northern Tasmania. In 1811 and 1812, Captain Barclay was the captain of this ship, the Providence. The Providence was privately owned at this point in time, and Captain Barclay's role was to basically, as in cargo shipping today, fill her up with cargo and get her to and from ports where more cargo can be found. And in 1811, he arrived in Sydney with pretty much the major cargo that was being brought into Australia at the time, prisoners of both sexes from Ireland. He spent the winter of 1811 in Port Jackson, and on the 20th of October 1811, he headed for China, and his cargo at that time was seal skins and soldiers. He arrived subsequently back in the UK on the 15th of September with a cargo of tea and porcelain china. The ship that he was on later became what's known as an East Indiaman, which is a ship that was involved in the trade, the two-way trade between, or three-way trade between India, China, and and England, and the primary items of trade there were tea and opium. But how is that relevant to Kiwi, you ask? Well, Barclay bought a skin of a kiwi from a sealer in Port Jackson in Sydney. And we can establish through the shipping records that kiwi was probably came from one of 6 vessels working in New Zealand and down on the subantarctic islands. And as I'll outline in a second, I believe that this kiwi probably came from a sealer on board the ship that's illustrated here, which is known as the Sydney Cove. This was an infamous ship that it was actually credited with starting the sealing wars in southern New Zealand and in Kōrero, but its history was certainly not one that one could take great pride in. Anyway, Captain Andrew Barclay returned to the England in 1812 with his kiwi. He subsequently gave that kiwi to a friend, an illustrator, William Evans, and Evans gave that kiwi to this man, this illustrated here, George Shaw of the British Museum. And George Shaw described that kiwi in a series of books that he was producing called the Naturalist's Miscellany. And sadly, he died shortly afterwards. The Naturalist's Miscellany is quite a rare series of books, and this, as well as a description that was in Latin, which gave this kiwi specimen the name Apteryx australis. It was illustrated here in these 2 illustrations, as the kiwi specimen was actually a skin. No one actually knew what kiwis actually looked like and how they actually stood, and the illustrator clearly thought that they looked or walked very much like modern penguins do today, and that's why this illustration is so grossly inaccurate. The the details that the illustrator made of the leg and the bill are remarkably accurate, and even showing the little vestigial wing that we know now that Kiwi have. The illustrator is actually very interesting herself. The illustrator was actually a woman, Elizabeth Nodder, and she took over her husband's business when he died, becoming a famous illustrator, and the quality, especially of her botanical illustrations, is very famous. Many of those engravings from the 19th century that you might see for sale are often Nodder's work. And she has not been, fully appreciated in the story. So when George Shaw died, even though he was the curator of the British Museum, he actually had a large private collection. And this was the era of rich people having large collections of natural history, especially collections that they could show off to their friends. So when Shaw died, his collection was sold at auction, and it was sold to this fellow here, Edward Smith Stanley, who later became the 13th Earl of Derby. He was a politician and a naturalist, and very richer than God, you might say. And this is his country seat, Knowsley Hall, which is just outside Liverpool. He had a vast collection of natural history and ethnology, and on his subsequent death, his collections were given to the Merseyside County Museum, and they became the core of the Liverpool Museum, which is now known as the Liverpool World Museum, and this is its building in central Liverpool. The curator, when we were visiting there, was a big fan of the Beatles, and she, in fact, was present during the very earliest shows of the Beatles in the cavern, and that was one of the reasons that we were very keen to be in Liverpool ourselves. So there's a Beatles collection to this earliest specimen of Kiwi. During 2017 and 2018, myself and my wife, Vanessa, visited our US and European museums as part of this Kiwi Rescue genetics project, and we examined more than 280 specimens of Kiwi to try and get an idea of locations that Kiwi were collected around New Zealand. And we were fortunate enough to get a sample of skin from the holotype of Apteryx australis that was held at Liverpool. And this is the actual specimen. So this is the actual specimen that was illustrated, that made its way to Sydney, probably had a quick trip to China, and then turned up in London in 1812. And it's in perfect condition. It's been stored and it's in absolutely pristine condition. And an interesting thing you can see from this illustration is that's a North Island brown kiwi beside this specimen, showing how much larger Tokoeka are than the southern kiwi. So we examined this, measured it, looked at the labels, and this is us working in Liverpool. And we brought it back, the sample back to New Zealand to be sequenced for its DNA. And this is where we got a big surprise, I guess. So when Shaw described the species, he didn't give a location because he didn't know where it had come from. In the 1850s, based probably on sort of an incomplete knowledge of sealing in the 19th century, George Gray, who was also at the British Museum, just basically said, I think it came from Dusky Sound. And this was quite important because this meant that if there was a different type of kiwi in southern Fiordland, it would own the name Apteryx australis. And in fact, that was so influential that when Walter Buller decided that the birds on Stewart Island looked different, his colleague named the Stewart Island kiwi Apteryx australis laurii, laurii being Walter Buller's middle name. And this that meant that for more than 150 years, we believed that Stewart Island birds were called Apteryx australis laurii, and southern Fiordland birds were called Apteryx australis australis. But this hypothesis about where the first bird was collected needed testing. And so using sophisticated techniques, Jamie Wood and Leah were able to extract virtually all the DNA from this bird's skin. And when we examined the mitochondrial DNA, it was very clear that the Stewart Island, that this holotype was most closely related to the Stewart Island birds. And when we looked at a huge portion of the nucleic DNA using a technique where we look at SNPs, which are just how the nucleic DNA differs, and we found also that the holotype was almost certainly collected on Stewart Island. So this made us then question where on earth the type locality was, and we can probably be quite confident that this bird was actually collected on Stewart Island. This was backed up by looking at other features. The problem with kiwis is they're really quite difficult to measure, but they do have a few morphological features that you can look at. So we looked at the number of scoots on the leg, and you can see here the holotype had 2 scoots, and that was very typical of Stewart Island birds, and not like birds from Haast, which you can see there in the top right-hand corner, and unlike all other types of kiwi. So Rakiura kiwi have generally 2 or 3 scoots on the front of their tarsometatarsals, whereas Haast Tokoeka generally have 6 or 7. And when we looked at the bill and other measurements, we found that also those measurements were very typical of Rakiura Stewart Island kiwi. So we published this result in a journal called Conservation Genetics, and our conclusions were that the holotype comes from Stewart Island. The name Apteryx australis laurii is what we call a junior synonym, so it's basically can no longer be used, because in fact if you were ever used describing a Tokoeka from Stewart Island, you have to call it Apteryx australis or Apteryx australis australis, and that also means that there are no other scientific names available, and thus all other Tokoeka, if we're going to describe them for science, require new names. And we're hoping, or we will be working with tangata whenua to suggest those names, so that each genetically distinct type of Tokoeka has a distinct name. And the next step in this project is, and this is well underway, is we're sampling right throughout the historic range of kiwi, including a great deal of samples from places where kiwi are now extinct. And this will enable us to confidently describe the extinct diversity, which will enable us to be much more confident about how distinct the living diversity is. And that will enable us to very shortly be able to confidently give names to robust genetic units. And in the Tokoeka area, we're particularly interested in the populations which were between extant populations, the extinct populations, to try and establish how many other extinct groups there were. And so, as I say, that work's well and truly underway. And in fact, the sequence DNA has been sent off to be sequenced, and we should have the results in the next couple of months. So thank you very much.
Hey, many thanks for that, Paul. That was absolutely fascinating. Weaving together all that history and biology and a bit of art and science in there as well. We've just got one question popped up so far, and that's good because we haven't got too much time. Do you think the holotype was potentially collected from Rakiura around the time of the Sealers' War?
Yes, sorry, I didn't go into that. Yes, so when we look at the Sealers' records, we can establish that only one boat, the Sydney Cove, was based on Rakiura in 1810 and 1811, and that was based in Perseverance Harbour. So we can be pretty confident that that is where that kiwi was collected. There's just a tiny chance that it was actually from an extinct population on the mainland, but the robustness of our genetic data strongly suggests that it was collected during the sealing wars on Rakiura.
Thanks. I've got one little question of my own. Might just take 1 minute. How many different Tokoeka do you think there will be when you've finished this exercise?
I believe that there will be 4 plus 2 extinct. The 2 extinct populations, one in essentially Southland and Otago and Canterbury, they're quite distinctive, and I think that they probably will deserve full species status. The genetic diversity of the ones, well, you know, the distance that each of the Tokoeka and Fiordland and Haast are from each other is still needs to be examined in greater detail, and that's what we're doing with this DNA work.
Great, thank you. Look, there's one final question, and we just don't have time to answer it. I just want to put it out there for everyone to think about, because we're going to come back next week with a further webinar around this same research program and diving into more science details around the DNA aspects of this program. So Jess has asked, what does this mean? What does all this mean for Kiwi conservation? So I think that might be a good parting sort of thought to leave with all of us, and I hope we'll be able to come back with a few insights into that as part of our next webinar next week. If you haven't signed up, the link to do so is on the invitation that you had for this seminar as well. So go back, check that out, and sign up to hear more in this exciting tale. But just for now, thanks very much, and we'll look forward to seeing you again next week. Bye.